So the RNA world. So we'll be seeing what is this RNA and what are the different types of RNAs are there. Okay. So we know RNA means it's a ribonucleic acid, which is directly involved in the protein synthesis. Yes or no? It is ribonucleic acid and it will be helpful for protein synthesis. Directly involved. Okay. So what is the major role of this RNA? It acts like a genetic material in certain viruses. It acts like a catalyst. It acts like a messenger in protein synthesis, right? So it will bring the information from the DNA. That information, the rRNA will use and it will synthesize the proteins. So these are the three important functions that RNA do. Genetic material, catalyst. What is meaning of catalyst? It will do the catalyst in the self, it's whatever that it is doing, the same thing. So it will be helpful for protein synthesis. So types of RNA. So first one is messenger RNA. So what does this messenger RNA will do? It will carry the message from the DNA. Okay, so whatever the DNA template is there, from that it will read the codon and it will synthesize the complementary codon. So that complementary codon will be helpful for making the proteins. So the message is present in the DNA, that DNA information, this mRNA will take, it will utilize for making the proteins. So it carries message from DNA. This is an important function that it will do. Okay. Sometimes what happen, these histrons, whatever the cistron means, it's the unit of gene. Remember, cistron. So what is meaning of cistron? Cistron means it's just a unit of gene. If that unit of gene, which will synthesize only one protein, we call it as monocistron. If it is containing multiple cistrons, that means multiple genes, that single unit, and it will synthesize multiple proteins, we call it as polycistron. So remember, monocistron means it is a single cistron. Whereas the polycistron means it is having multiple cistrons. So multiple cistrons means multiple proteins. So the unit of the RNA, mRNA, that can give multiple proteins, it is polycistron. So where does this polycistron, it will happen? It will happen in the prokaryotes. Remember this. So only in the bacteria, you will be seeing the polycistronic nature. Whereas the monocistronic, it will happen in the eukaryotes. So remember, in eukaryotes, only we will be having the single cistron. In the prokaryotes, we will be having the multiple cistrons. Is this clear? So monocistronic means from single unit of RNA, mRNA, it will give single protein. Polycistronic means from single unit of RNA, mRNA, you will be getting different cistrons. That means different proteins, different genes. Understood? Is this clear?
From single mRNA, you'll get single set of protein, that is cistron. Right?
Monocistronic. No.
Okay.
Cistronic means the unit of gene.
Unit of, okay.
Yeah. So see, if you see this much of this mRNA and you are getting only one gene here. But if you see here, you are getting polycistrons. That's a multiple proteins from single set of mRNA. So what here we say is from the DNA, only one unit of gene we can do at a time. So that's called monocistronic. Whereas from the DNA, you are making a mRNA. That mRNA can give rise to multiple proteins. Then we call it as polycistronic at a time.
Yeah, got it.
Okay. So types of tRNA. So what does the tRNA will do? It is acting like a porter guide. Remember this. Okay. So what it will do? E will carry or E carries correct amino acid to protein site or site of protein synthesis. What is meaning of site of protein synthesis? Where does it will happen? Where does the protein synthesis will happen? Come on, where does it will happen?
Ribosomes.
Yeah, it will happen at the ribosomes. Ribosomes are called as the factory of the protein synthesis. So the amino acids are present in the cytoplasm. From the cytoplasm, you need to carry the amino acids to build this protein chain. So that is done by this tRNA. Okay, just do one thing. I'll tell you Uh, did I tell you before what is the use of different mRNAs? Different RNAs?
Sir, you, in the fourth PDF you did, sir.
Like what
Like,
is the function of hnRNA, mRNA, everything?
yeah, sir, we spoke about tRNA, and you gave two sentences about it, and
snRNA?
mRNA. No, 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 sir.
Okay, do one thing. I'll just explain you like a kind of small thing that will tell about what is the different RNAs will do. And then we'll talk about each of them in detail. So let's take a cell. So inside the cell, we have a nucleus. So inside the nucleus, we have all the genetic material. That's a DNA. So remember, this is DNA. Okay. So from this DNA, we know what it will come. There is a mRNA, it will come. Okay. Not only just mRNA, actually, there is another thing that will come first is the hnRNA. Just remember, from the DNA, you will get first the hnRNA. hnRNA is like the raw one. So what is meaning of raw one? First, suppose you take the crude oil. Okay. So what happens with the crude oil? It will be containing the dirt as well as the useful thing, right? Useful thing in the sense the petrol, diesel oil, and the dirt in the sense the waste oil. So the same way from the DNA, when you synthesize the RNA, it will contain the genetic information as well as the junk part. Understood? hnRNA means it will be containing both the genetic information as well as junk part. Remember this. So genetic information and the junk part. So what is this genetic information? We call it as the exons. Remember, these are called as the exons. And what is this junk part? It is called as the introns. Why we call it as the junk part? Because they don't code for any protein. Okay, they are just useless stuff. Whereas the genetic information that is actually present in the exons. Okay, those are the series of nucleotides which will code for an amino acid. We call it as exon. The junk part doesn't code for any amino acids or it doesn't code for any protein. Okay, understood? Remember, hnRNA, it will contain the series of exons and the introns. Now, what we need to do? We don't want to have this junk part, right? So what we'll do after crude oil is extracted, we will filter it and we will extract the petrol from that crude oil. The same way, we will filter out all this intron and I will just extract the exons. So that's how the hnRNA, now it will turn out to be the mRNA. There will be certain post-transcriptional events will happen that will finally convert the hnRNA into the functional mRNA. Okay. So here the post-transcriptional events will happen. So all this will comes under here. So genetic information, exons, remove these introns, keep the exons. So this is post-transcriptional events. We'll discuss today what is this. So just remember these are post-transcriptional events. So once this mRNA is formed, I'm not writing the hnRNA or mRNA. Directly I'm writing the mRNA here. Okay. 1 second, guys. Okay, from this DNA, now I got the mRNA. Okay, remember the mRNA is also present inside the nucleus. The hnRNA is also present inside the nucleus. But who will be helpful for converting this hnRNA to mRNA? So this is hnRNA. This is mRNA. Who will do this? Keep the exons and remove the introns. That is done by another kind of RNA that we call it as snRNA. Remember this. So who will remove these introns, the junk part? It is done by snRNA. So what is this snRNA? Small nucleotide RNA. What is hnRNA? Heterogeneous RNA. Why we call it as hetero? Because it is containing the exons and introns. So there are two different things. So we call it as heterogeneous.
Can you repeat what is snRNA?
snRNA, it acts like a scissors. Okay. So what does it will do? It will cut down all the introns and it will keep the exons. First, suppose, let me show you. So this is exon. This is exon. Let me show you here. So this is exon. So remember here, this is the exon, this is the intron, this is the exon, this is the exon, this is the intron, this is the exon. So you remove these introns and then you form only the exons. So you keep all the exons together. Okay, so all these are called as this one is called as the HNRNA. Why? Because it is containing the mixture of exons and the introns. HNRNA, we called it as heterogeneous because it is containing both exons and introns. Whereas now only the exons were present. So now we call this as the mRNA. To remove these introns and to keep all the exons together, for that we need a kind of scissor which will cut down these introns and which will join all the exons. That is done by the snRNA, small nucleotide RNA. Okay, so actually the small nucleotide RNA, it will be present with the proteins, and we call this combination as the spliceosomes. If you read, it will be written somewhere in the textbook. We call them as the spliceosomes. Okay, what is spliceosomes? There's an RNA plus proteins, like the kind of ribosomes. What are ribosomes? We know ribosomes means the RNA, rRNA plus proteins. Yes or no? rRNA plus proteins. We call it as the ribosomes. What does this ribosomes will do? It will catalyze the protein synthesis. The same way the spliceosomes, which is snRNA plus proteins, it will be helpful for removing all the junk part, all the introns, and join these exons. That will become the mRNA, which is a functional mRNA. Now, if the question comes, which are all the RNAs that are present inside the nucleus? The hnRNA is present inside the nucleus. snRNA is present inside the nucleus. Even the mRNA is present inside the nucleus. Understood? Is that
Yes, sir.
clear?
Yes, sir.
Okay. Now, this mRNA, it will just go out of the nucleus. It will travel through the endoplasmic reticulum. And we know this endoplasmic reticulum, they contain the ribosomes. What are ribosomes? Simply rRNA with certain proteins. We call it as the ribosomes, the rough endoplasmic reticulum, okay, which contains the ribosomes. In these ribosomes, we have another kind of RNA, that is rRNA, ribosomal RNA. Okay, this ribosomal RNA, what it will do? It will read the mRNA. Whatever the information that brought, it will read the mRNA. Okay, and he will tell that tRNA to bring those amino acids to the protein site. So this tRNA, he will act like a porter guy. So what he will do? tRNA, in the sense, it is called the transfer RNA. He will be acting like the delivery guy. He will collect all the amino acids and he will bring to the protein site, to the ribosomes. Now you will be making the protein or amino acid chain, the peptide chain. Is that clear? So total, how many RNAs are there? One. The first
Only one
format is,
second.
here the tRNA will act like a porter guy. He will bring these amino acids to the protein site. Okay. So just remember, rRNA, he will read the mRNA, whatever the information that messenger RNA brought, and he will tell the tRNA, okay, you go and bring this particular amino acid. Okay. Whenever he reads a amino acid, then he will tell that I'll tell you in the translation process. But just now understand what are the different types. Okay. So you will bring the information, that information will be read by the rRNA. You can tell the rRNA is like an engineer. He will read those data and he will ask the tRNA to bring those to bring those amino acids. From that, they will make a chain of amino acids, the peptide chain.
Okay.
Understood? Is that clear?
So, from the nucleus, the mRNA will go out. So
Yeah.
it will take the exons. So when it takes out the exons, the rRNA will read those exons and the information, and it will tell the tRNA to set for a set of amino acids.
To bring the amino acid. Each tRNA will bring one amino acid after reading one codon. That we'll tell in discuss. I'm just telling in brief. Actually, the detailed function I'll tell you. Just remember like that. Okay. So now what are the different types of RNAs are there? One, the HNRNA. That is the one which is formed first. So this HNRNA with the help of the snRNA, it will make the mRNA. So this is 3. Now this mRNA will come out of the nucleus. So remember 1, 2 and 3, they are present inside the nucleus. But mRNA is present both inside the nucleus as well as in the cytoplasm. Remember this. The question can come like that. So which of all the RNAs present inside the nucleus? That means HNRNA, snRNA, mRNA. Which of the RNA present both inside the nucleus as well as out of the nucleus? mRNA. Which are present only outside the nucleus? tRNA and snRNA. Sorry, tRNA and rRNA. Only outside the nucleus. Understood? Is that clear?
Yes, sir.
So you can remember like hnRNA is like a raw RNA, mRNA is like the modified
Yes,
functional
sir.
messenger RNA. Who will convert this raw to the modified? It is the snRNA. Now this modified RNA will come out of the nucleus. It will go to the protein synthesis site where the engineer is present, that is rRNA. Now this rRNA will read the information brought by the mRNA and he will tell the porter guy, the tRNA, to bring the particular amino acid. So he will bring the particular amino acid to the protein site. Now they make the protein chain. Understood? So this all will happen in the endoplasmic reticulum on the ribosomes. Now this protein chain is formed that will go for packing to the Golgi apparatus. If you remember, there is a Golgi apparatus will be present. So these Golgi apparatus, they will make packaging of this. And this, they will be released like a lysosomes. The lysosomes will contain all these proteins. So they can do the endosmosis, exosmosis, and they will send this information out of the cell as well. Clear? That's all different thing. We're not going into depth. Understood? What is the different types of RNAs and where they present? Is that clear or any doubts?
Yes.
Understood, sir.
Okay. So this is what the tRNA will do. Okay. So carries correct amino acid to the site of protein synthesis. That's the ribosomes. And he is having the clover leaf shaped structure. We'll talk in more detail about this tRNA before we start the translation. Okay. Next one. rRNA is just present inside the ribosomes. It will catalyze protein synthesis. Understood? So we will synthesize the proteins. And this is containing the larger subunit and small subunit. Remember, when these large subunit and small subunit present separately, then it is for translation, it is inactive. Remember, this is inactive when they are present separate. This is active when they are together. Okay, so translation, active translation is inactive. That means translation will not happen. So there will be no translation when they are present separate. Understood?
Yes, sir.
Yes.
So what is the difference between the replication and transcription? See here, replication will be carrying all the entire DNA for making another copy of DNA, right? But transcription is not like that. Only a small segment of DNA is read, and from that you are making that mRNA. From that mRNA, you will be getting only one protein, then you will call it as monocystronic. But if you are getting multiple proteins, then you call it as polycystronic. Understood? Whereas here both the strands are copied, here only one strand is copied. And I told you what is the reason for copying only one strand. If both the strands are copied, then it will make the double strand, and those double strands will bind each other and it will make translation will not happen after that. And the second thing, from the single strand, you are getting two different kinds of proteins. So that's complicating the entire genomic process. Yes or no? So the nature has selected only one strand to be copied. Next one, replication needs the DNA polymerase, DNA dependent DNA polymerase, whereas transcription requires DNA dependent RNA polymerase. Okay, so this will come here. So this is the DNA dependent RNA polymerase. And here for replication, the primers are needed because the DNA dependent DNA polymerase cannot start the transcription. Why? For this, there is a hydroxyl group is needed at the third end. Then only it can do it. But if there is no OH group, then you cannot do. So that OH group will be formed by the primer. Understood? So you will be synthesizing a small fragment of the RNA. That RNA will be having the third end, and this third end is containing the OH group. So to this OH group, the RNA, the DNA polymerase, it can bind and it can continue the transcription. So this will be the RNA, this will be the DNA. Later, this RNA will detach and it will be made, it will be continued by the DNA. So the DNA will be added here. Understood? Here the product is the double strand, here the product is a single strand. And for this, the primers are not required. And remember, this primer is made by the primase enzyme, RNA primase enzyme. Clear? Any doubts? The difference between the replication and transcription? These two processes is clear for you?
Yes, sir.
Yes, sir.
And whatever the tools required to copy the DNA to RNA for transcription is DNA dependent RNA polymerase, DNA template, and the nucleotides. Okay. And the structural gene, promoter, and the terminator sequence. So if you see the transcription unit, so these are the transcription unit. So these are required tools. These are the transcription thing. So first the promoter, then the terminator sequence, then the structural gene. Promoter is present on the upstream of the coding strand, and terminator is present on the downstream of the coding strand. Whereas the structural gene present between promoter and the terminator. And remember, before promoter, whatever the region is there, that is UTRs. So for suppose you see, so this is the promoter region. This is the structural gene, and this is the terminator. And whatever the area present before the promoter gene and after the terminator gene, so we call them as the UTR, untranslated regions. They are called as untranslated regions. Understood? That means they cannot be translated because we know the promoter region, before that there is no translation. Sorry, transcription. Sorry, this is not untranslated. This is untranscripted region. And after the terminator, you have no untranscripted regions. Clear?
Yes, sir.
So if you see,
Yeah.
this is the coding strand. If you take it like a coding strand, then this will be the upstream and this will be the downstream. This will be the fifth end. This will be the third end. Yeah, understood. So
Yes.
just give me a second. So just revision. So initiation, that's a, you know, right? So DNA polymerase along with the sigma factor, it will attach to the DNA molecule. It will recognize where the promoter is. So who will help for recognizing that promoter region? The sigma factor. Remember this. Without sigma factor, we cannot do that. So next one, the DNA double helix unwinds, exposing the DNA template strand to form the new RNA. Is that clear? So remember this. So remember, one is this one, the sigma factors, and the other one is the promoter thing. Okay. Next one, elongation. Elongation is present between the, sorry, elongation is the next one. So what happens? When do we tell that elongation has begun? When the sigma factor will leave off. Okay. The RNA polymerase now enter into the structural gene. So where it will synthesize the protein, sorry, where it will synthesize the mRNA. Okay. Where you will be seeing the growth of mRNA. And finally, the terminator. So now this rho factor, it will attach to the RNA and it will climb over it and it will reach the point where the DNA and RNA bond. Okay, so it will unwind the RNA from the DNA and it will release the RNA, and then the RNA polymerase will also be released, where the transcription process will end. Understood? So release of the RNA from RNA polymerase, it will be done by the rho factor. Is that clear?
Yes, sir.
And we saw what are the types of RNAs. So remember mRNA, what is the location where mRNA is present?
Both outside the nucleus and inside the nucleus.
Yes, sir.
Yeah. So remember that one. Okay. So it is present in cytoplasm
And
and
nucleus.
nucleus. Whereas the tRNA, it is present in the
Outside the
cytoplasm, outside the nucleus. And rRNA, it is also in the cytoplasm.
Got it.
Okay. So that's it. Where the for eukaryotic transcription and the prokaryotic transcription, this is common thing: initiation, elongation, termination. Okay. But if you come to the eukaryotic transcription, there is further complex thing will happen, that is splicing, capping, and tailing, which will not happen in the prokaryotes, which only happens in the eukaryotes. Okay. So remember, this thing only happens in eukaryotes. It will not happen in prokaryotes. Remember this. Is that clear? So we'll see what is that post-transcriptional modification, or you can say how the hnRNA is converted into the mRNA. So few things about the eukaryotic transcription you should notice. So first one, it is more complex compared to the prokaryotic transcription. This is one thing. Why? The prokaryotic transcription will not have the post-transcriptional events. Only the eukaryotes will have. So that's why it is more complex. Understood? So the second thing, where does this eukaryotic transcription will happen? I mean, the transcription and translation will happen. Generally, this occurs in G1 and G2 phases of cell cycle. If you remember the Mrudula, we have discussed the G1 and G2. What happens in the G1, G2? G1, it will be helpful for synthesizing certain proteins. Those proteins are required for the next phase, which is S phase. Proteins that are required, or the enzymes that are required for the S phase, it will be synthesized in the G1 phase. What happens in the G2 phase? It will synthesize the proteins that are required for the M phase, or you can say the actual cell division phase. So where actual cell division will happen. So in that. Understood? So remember, so in G1 and G2, you will be seeing the transcription and translation. Not only in G2, you will be also seeing in G1. So in G1, G2, eukaryotic transcription, where this happens. Next one, where does the transcription happens? So transcription happens inside the nucleus. So transcription happens inside the nucleus. So with the help of this transcription inside the nucleus, you will be forming the mRNA, hnRNA, rRNA, snRNA, tRNA. All these are synthesized inside the nucleus. Now they will come to their respective places after the transcription is over. Understood? And we saw which polymerase enzymes will do the different types of RNAs. Like polymerase 1, it will synthesize the rRNA. Polymerase 2, it will synthesize the hnRNA and mRNA. Polymerase 3, it will synthesize the snRNA, tRNA, and 5S rRNA, right? So all those transcription will happen inside the nucleus. But not only there is other places where the transcription can also happen, that is, and it also happens inside the mitochondria and chloroplast. Can someone guess why it happens? What is the reason mitochondria and chloroplast, these organelles, they can do that?
The mitochondria to produce more energy and all that, it requires transcription.
It's okay, fine. Then what? Chloroplast will do the photosynthesis, so it is producing the transcription. Not that, because they have their own genetic material. So have their own DNA. Can you tell me why these organelles have their own DNA? Because if you see the evolution, actually these are the prokaryotic cells. small prokaryotic cells. These small prokaryotic cells, it is engulfed by the big prokaryotic cell. So they entered into the big prokaryotic cell. Now this big prokaryotic cell, it is not digested these small prokaryotic cells. Instead, it kept them alive. It kept them inside safely. And what happened? They provided the energy. Okay. So what these small ones did, so they generated the energy by using whatever the food resources the big one is giving. So that's how over a time this become the cell part, and that's how mitochondria is created. The same way with the chloroplast, the other small one which entered inside and it started utilizing the sunlight and it started producing the energy. So that's how it became the part of the cell organelle. Now the prokaryotic cell is turned into the eukaryotic cell over the time of or over the course of evolution. Understood? So that's how eukaryotes are formed from the prokaryotes. And where the mitochondria and chloroplast, which used to be a prokaryotic cell, they become the cell organelles. So they have their own DNA. So that's how they are almost similar to the prokaryotic cell. Remember this. So they can do their own transcription and translation process. So transcription can happen inside the nucleus, and it also can happen inside the mitochondria and chloroplast, because they have their genetic information. Understood? They have their own DNA.
Yes, sir.
So next one. And remember, each of the, if you see in the prokaryotes, all the types of RNAs, it is produced by only one type of polymerase. That is only RNA polymerase. Whereas in the eukaryotes, there are different types of RNA polymerase are there. Okay. So just remember, different RNAs synthesized by different polymerase. Clear? So different RNAs synthesized by different polymerases. This you need to remember. And if you see, where does the transcription is happening? Where is it happening? Transcription, where it is happening?
Sir.
Inside the nucleus. Yes.
Yes, sir.
And where does the translation is happening? Where does the translation is happening? In the ribosomes, right? Which is present in the cytoplasm. So it's happening inside the ribosomes. Now, if you see, the site of the transcription is different and the site of the translation is different. Now, whatever after transcription, whatever the mRNA that is formed, it takes time to go from the nucleus to reach the ribosomes. Yes or no? So that mRNA, which is produced inside the nucleus, it will just go out of the nucleus, passing through the nuclear membrane, and it has to pass through the endoplasmic reticulum and finally reach the ribosomes, because they are at the different locations. So remember, in eukaryotes, transcription is not, the translation is not immediately followed after the transcription. Or you can say the transcription is not immediately followed by the translation. Clear? Remember this. So as they are in different locations, mRNA takes time to pass from nucleus to ribosomes. So the transcription and translation will occur at different time period. Okay, so you can say transcription is not immediately followed by translation. Why? Because they have a different sites. They occur at different sites. Understood? Is the same with the prokaryotes? Tell me, is that the same with the prokaryotes?
Sir, it all happens in the cytoplasm only, right?
Yeah. So prokaryotes, where the transcription translation happens simultaneously. Remember that thing. Okay. So prokaryotes, transcription, translation happens simultaneously. Understood? So this is only for the eukaryotes. This is only for eukaryotes. This is only for eukaryotes. This is for the prokaryotes. Is that clear?
Yes,
Yes,
sir.
sir.
Yeah. One more thing. Whatever the mRNA that is produced here or the hnRNA which is produced, it is very unstable. Okay. So instability of this mRNA. So whatever this mRNA that is produced or hnRNA which is produced, it is very unstable. Why? Because whatever the head and tail is there of this mRNA, they can be easily attacked by the enzymes present. Okay. So instability of this mRNA due to enzymatic attack. That means the enzymes attack makes the mRNA very unstable. So makes mRNA very unstable. So what do we do now? So this is making very unstable. So we need to modify this. Okay. For that, we will be doing the post-transcriptional things. So we are doing the capping and tailing. Okay. So what you will do, that will be you are putting the helmet, the cap, so that it will prevent the enzyme to attack on the head. And the same way the tail, you are doing this tailing so that the tail is protected or the bottom is protected. So it will prevent the enzyme to attack at the bottom. So you are doing capping and tailing, which will happen in the post-transcriptional events. The same way, I mean, before capping and tailing, the one more thing that will happen is the splicing. One more thing that will happen is the splicing. So what is this splicing? I told you there is a junk part in the DNA as well. Okay, we need to remove that junk part. So first splicing will happen, then capping and tailing will happen. So this is second, this is first. Remember this. So splicing, what happens? Where whatever the hnRNA is there, that hnRNA has to be converted to the mRNA. Okay. And this mRNA, after the capping and tailing, it will fully become the functional mRNA. So this is the splicing, where you will be using the spliceosome, that is snRNA plus proteins, to convert this hnRNA to mRNA. So how do you convert? Whatever the non-coding regions are there, you will be removing it. And you keep or you join all the coding regions. So that's how it will become mRNA. Understood?
Yes, sir.
What about you, Mrudula? Any doubts?
No doubt, sir.
So let's see in detail what are these post-transcriptional modifications. One second. Okay. So post-transcriptional modification, this is very, very crucial for the eukaryotic cells. Okay. So whatever the primary RNA transcript that is formed. So can you say what is that primary transcript? Primary transcript. That is nothing but the hnRNA, which is formed by this post-transcriptional modification. You will convert that into a functional RNA molecule. Understood? So
Yes, sir.
remember this post-transcriptional modification, it will happen after the transcription and before the translation. Okay, this should happen because this will very helpful for gene expression and regulation. Okay, without this, that couldn't happen. So let's see what is this splicing, capping, and tailing. So first one is the RNA splicing. Remember, there's no DNJ splicing, it's a RNJ splicing. So this is the most complex post-transcriptional modification. Okay. So what happens here? The removal of the non-coding introns. The main thing that will happen here, removal of non-coding introns. So remember, this is a non-coding part, and this is a exon is a coding part. So you will be removing all these introns. Whatever the non-coding part is there, you will remove, and you will join all the coding part. Okay, so that we call it as the splicing. Understood? And this splicing is done by, or is it carried out by, the spliceosomes with the help of spliceosomes. Can you tell me what is the spliceosomes?
SNRNA plus protein.
Yeah, spliceosomes are nothing but snRNA plus proteins. Remember this. So removes the non-coding part, that is the introns, and we keep all the coding parts together. Okay, is that clear? What happened here?
Yes.
Yes, sir.
And remember, whatever, sometimes they can call the hnRNA, or sometimes they can call this primary transcript. But remember, this primary transcript or hnRNA, both will be containing introns plus exons. And the functional RNA, which actually only contains the exons. Okay, so what is this introns? Introns, these are the non-coding or non-functional or intervening sequence. They're just present like that. They don't do any function. Whereas what is exons? Coding or functional sequence in RNA, which codes for a particular protein. But some scientists, they believe that these introns, they are not so useless. I mean, they are not junk part. Actually, they have a, I mean, before earlier, they was used to think this is only the junk part, but they did many research and found that actually this intron is a protecting material. For suppose, just remember like this. For suppose, let's take a, what do we say? Yeah, let's take like a female killing. Okay, there are some, yeah, you might have seen this one. What is it? The recent movie, Kalki movie. If you watch, in that movie, what happened? If the king, I mean, whoever the supreme, if he finds any girls, he will take out. Okay, so what happening then? They, whatever the girls they are there, they were just present like the boys to protect themselves, right? So that they can hidden in between these boys, so they cannot be found. The same way, for suppose if you have the genetic information, in that genetic information, if you have the DNA actually 99% it's just a junk, and only in that 1% actually codes for genes. So remember, whatever the 2 meters length of DNA is there, it's actually that entire length is just a junk. Only this 1% is actually codes for the protein. So that what happened, this 1% is just scatteredly present along this 99%. So if something due to the mutation or due to the chemical damage or due to the environmental exposure, if the DNA gets damaged, there is a very less chance for this 1% to get damaged. And whatever the 99% is there, that will be damaged, but not this 1%. Understood? So it's just like a protecting. So that's what the introns do. That's one version. The second version they do is, so these introns, exons, introns, they are just present like that randomly. Okay. So what happens if I change this intron to some other location? So this will change the nucleotides sequence. So changing this nucleotide sequence, it will bring a small change. That small change will be helpful for evolution. Remember this. So it is helping to contribute the, it is helping by changing the genetic information, which is playing a very important role in evolution. Understood? The second thing is, So it is present in the abundant of junk so that whatever the main gene part is there, it will not be scrutinized by any external factors. Understood? So what's the use of these introns? This is not needed, just for your information. I'm just saying it.
So this
Understood?
is why in a human genome project, they told we'll have the both introns and exons like that, right?
Yeah. We'll discuss that. That's the last part of this chapter.
Yes.
Okay. So understood. What is this introns? What is this exons? What are the use of the introns and the exons?
Yes, sir.
Yes.
Okay. So see here. So here the splicing will remove all the introns. So splicing, that is a removal of introns and join the exons. Okay. And remember this splicing, it is catalyzed by a large protein complex. What is that? It is catalyzed by large protein complex. What is that called? Spliceosome, which is made up of protein plus RNA. Understood?
Yes, sir.
It's an enzyme now, sir.
Yeah, it's an enzyme. Okay, so it's a protein complex. So you see here, the snRNA, sorry, the introns were removed and all these exons were joined. Next one, capping. It is the addition of unusual nucleotide on the fifth end of the mRNA. 1 second. So what is capping?
So methyl group will be added in the promoter.
Yeah. Okay. So it is the addition of, remember this word, unusual. That means it's a completely unique thing, right? It's not a normal thing. So why is it like that? Because you will be adding the series of 7 methylated guanosines. Okay. You will be adding 7 methylated guanosine triphosphates at the fifth end. Remember this. Where it is happening is also very, very important. This is very, very important. So unusual nucleotide sequence, that is at the fifth end, you will be adding 7 methylated. That means they are joined with methylated. Okay. They are just having this methyl group. What is methyl? CH3 group. So guanosine triphosphate. This is not monophosphate, this is a triphosphate, remember. So which is also called as MGPPP. Here PPPP in the sense triphosphates. Okay, or you can say like GTP as well. Methylated guanosine triphosphate. Understood? Which is a unusual nucleotide. Why it said is unusual is actually, for suppose they are present this way. That means third and fifth end they are present like this. The other nucleotides, if they are present like thymine, adenine, guanine, cytosine, sorry, uracil, let's say, adenine. If they are present like this, all are in this direction. Whereas this MGPP, it will be present in the opposite direction and which will be attached at the fifth end. So that's why it's called as the unusual nucleotide. This is not needed. Just understand why it is called the unusual. Okay. Just I'm telling, but that's a very depth thing you don't need to know. Is that clear?
Yes, sir.
Yes,
So
sir.
remember, where does the cap is present? It is present on the fifth. Okay. Remember this thing. So the cap, it is present on the fifth thing. And what does this cap will do? It will protect the mRNA. So whatever this cap is there, it will protect the mRNA, or this, whatever the hnRNA, which is converting into mRNA, that is, it will protect that. Okay. And what else it will do? It will also help for marking, like, I mean, there will be kind of antenna thing. Okay, not antenna, I'm just telling like example antenna thing. Okay, so which will be facilitating the export of this mRNA to the nucleus, to the cytoplasm. Okay, so from the nucleus to the cytoplasm. So where are these present? These are present inside the nucleus, right? They need to know their way. It acts like a GPS. For suppose if you want to travel from one location to other location, how you are traveling? Using the map. The same way, they will be having some signaling thing. That signaling thing is provided by this cap, so that they know what is the way from going to nucleus to the ribosomes. Understood? Remember this. So facilitate export from nucleus to the cytoplasm. That's it. That's one more thing that it will do. And what else it will do? Ribosomes, when these things are coming, like when the mRNA is coming, the ribosomes has to receive, yes or no, so that they will do the translation. The ribosomes cannot recognize these if the capping is not there. Okay, see, whatever these 3 points, it's not there in the NCRT, it's much more deeper, but I'm just telling you. Okay, the third function that it will do is it will be helpful for ribosomes to recognize. Okay, so to recognize the mRNA for translation. Okay, for translation initiation. So these are the three important functions. That's why we call it as functional mRNA. Okay, if it is not well developed, then it will not be utilized. Okay, the ribosomes cannot receive them. They will not go to the ribosomes. Instead, they can go to some other places. If they are not, if there is anything disorder is seen. Let me put charging. Understood? What is the use of capping?
Yes. Yes, sir.
So these are the only complex things that only happens in the eukaryotes, not in the prokaryotes. I mean, similar kind of things will happen, but not this much complex. Prokaryotes also need to have these signaling things because they also need to reach the ribosomes. So for them, it will happen in different way. So that's about the capping. Any doubts?
No doubt, sir.
Yes, sir.
Okay. So the other one is the tailing. So we call this as a tailing, or sometimes they can also call it as the third end polyadenylation. Don't confuse if something is given like this. Okay, so polyadenylation means you are adding a large number of polyadenylate at the end of the, or you can add at the tail region, or you can add at the third end region. Understood? So remember, at the third end of the mRNA, there will be a lot of polyadenylated tails will be added. Those are the all adenosine nucleotides. Okay, so all these are added. That we call it as polyadenylation. This will happen at the third end. Remember this. This is very, very important. So capping will happen at the fifth end. This is the fifth end. This is the third end. Tailing will happen at the third end. Understood? So all these are nothing but poly A. And remember, whatever the A we are adding here, it is a adenosine monophosphate. See, these are all for capping. You are adding methylated guanosine triphosphates. These are triphosphate. Whereas here, you are adding monophosphate. Remember this thing. Okay. How many you will be adding? Some 200 to 300. 200 to 300 is AMPs, adenosine monophosphates, you will be adding. Is that clear?
Yes, sir.
So it will be between 200 to 300, or you can say the average of 250 will be added at the third end. So what does it will do? This is also extra information, just know it. So it will enhance the mRNA function, or you can say the mRNA stability by protecting from degrading enzymes. Okay, and also it helps in aiding the translation, as well as this also acts like antenna, which will be helpful for signaling. So it will show the path from the nucleus to the cytoplasm. Understood? So remember, by this, they will be helpful for these modification all will be helpful for regulation of this gene expression, the quality, as well as, I mean, there will be some stress responses as well. For suppose sometimes you need the quick mRNAs and all for the body to respond. For that also they are needed. Those all you don't need to remember. So remember, after all these modifications, finally the hnRNA will become the functional mRNA. Okay, so hnRNA will finally now become functional mature mRNA. Now this mRNA, it is ready to leave the nucleus. Okay, so leaves nucleus. Remember, where does the post-transcriptional things will happen? It will happen inside the nucleus. Okay, so it will leave the nucleus and it will go to the cytoplasm. So what are the advantages? So capping and tailing, the main role for them is to protect transcript. That is nothing but the hnRNA, or you can say the primary transcript from enzymes. Second thing, so these modifications help RNA molecule be recognized by molecules that mediate RNA translation. RNA translation to proteins. Is it clear? So this will be the tail. This will be the cap. You can say this is the fifth end, this is the third end. So that's it. This is the complete detail about the transcription. So we're done with the replication, transcription is done. So we finally left with the translation. And then regulation part is there. So remember, so what's the difference between the prokaryotic and eukaryotic transcription? Prokaryotic transcription, it will happen inside the cytoplasm, whereas here it happens inside the nucleus. Remember this. So it happens in the cytoplasm. This will happen inside the nucleus. Here only one RNA polymerase is there that will synthesize all the types of RNAs, whereas here 3 RNA polymerases are there. And here there is a need for the sigma factor for initiation. It doesn't needs transcription factors, or you can say the sigma factor. Okay, so RNA formed is polycistronic, whereas here it is monocistronic. So single protein, here multiple proteins. No post-transcriptional, no modification in the sense, no post-transcriptional events. Whereas here post-transcriptional modifications are seen. Difference between the replication and transcription, entire genomic DNA. Okay, I think we have discussed all this. So any doubts?
No doubt.